Throughout human history, diseases caused by pathogenic microorganisms such as plague, cholera, and severe acute respiratory syndrome, coronavirus, SARS-CoV have killed millions of people. Do you remember getting vaccinated during the COVID outbreak? In many ways, vaccination is closely related to the use of microorganisms. To make a vaccine, the microorganisms are either weakened or killed with heat or chemical. The genetic materials of microorganisms is also used to make DNA vaccines. Microorganisms are also used as a vaccine carriers to enhance the immune response and memory against disease-causing pathogens. How do vaccines work? Vaccine helps the body identify microorganisms that causes disease by stimulating an infection. So, when a pathogen enters the body, it triggers an immune reaction.
The first generation vaccine are manufactured by growing leaf vaccinia virus in the skin of leaf animals. Vaccination results in a skin lesion that fills with pus and eventually cracks over. After 2 to 3 weeks, the scalp will fall off and leave behind a vaccine scar. The second generation vaccines are also administered through scarification with a bifurcated needle, and they carry the same side effect as the first generation vaccinia strain that was cloned. However, the use of eggs or cell culture allows for vaccine production in a sterile environment. Ernest William Goodpasture, Alice Miles Woodruff, and G. John Bading grew vaccinia virus on the chorioallantoid membrane of chicken embryos in 1932. The third generation vaccines are based on attenuated vaccinia viruses that are much less virulent and carry lesser side effects. The attenuated viruses may be replicating or non-replicating. Modified Vaccinia Ankara is a replication incompetent variant of vaccinia that was developed in West Germany through serial passage. After 572 serial passages, the vaccinia virus had lost over 14% of its genome and could no longer replicate in human cells.
What are the criteria of a good vaccine? Ideal vaccine should have a high temperature stability, especially for usage in tropical nations, eliminating the need for pricey cold chains. Next is cost. A vaccine must cost less than 50 cents per dosage in order to be included in the WHO expanded program for immunization. Currently, UNICEF provides the 6 infants immunization included in the worldwide program for a cost of roughly 30% each schedule of injections. This provides some insight into the benefit of bulk purchase and is far less expensive than the identical vaccines offered in several national programs. Furthermore, enduring immunity. After 1 or 2 doses, several attenuated live virus vaccination, such as the measles and vaccinia vaccines, provide long-lasting immunity. Most non-infectious vaccination typically need a series of administrations with adjuvant to produce outcomes that are comparable. There is now a lot of research being done to look for ways to cut down on administration, like the usage of controlled release preparation.